Hello and welcome everybody to the chapter Mscdfsm Biochemistry, Digestion, Absorption and Transport of Carbohydrate, Proteins and Lipids. What are the basics of this chapter is? First, we have to know what is digestion. Digestion is the process by which nutrients, namely carbohydrate, protein, lipids are converted to simple sugars, amino acids, fatty acid and glycerols are called digestion. This process is facilitated by secretions of various glands and organs like salivary glands, stomach, pancreas, liver, hormonal and neural control regulate the secretions. Microvilli of intestine are the major site for absorption. What is absorption? It involves the transfer of materials through mucosa of alimentary canal, blood and lymph vessels. Digestion. Digestion is carried out with the help of enzyme and large molecule break down into smaller ones. Protein break down into amino acid, carbohydrate into monosaccharides and fat into fatty acid and glycerol and monoacyl glycerols. Vitamin, mineral, water, cholesterol are not broken down before absorption. Digestive system. It is coiled muscular 6 to 9 meters long when fully extended from mouth to anus. Various parts are mouth, pharynx, esophagus, stomach, small intestine, large intestine and anus. Digestive system is made up of two group of organs. One is alimentary canal, another one organs. Alimentary canal. It includes mouth, pharynx, esophagus, stomach, small intestine, large intestine, appendix, rectum, anal canal. Accessory organs are tongue, teeth, salivary gland, liver, gallbladder and pancreas. Digestive system ingest food material, digest and absorb these products and eliminate unusable materials. Digestion and mouth. Mouth has tongue, teeth, salivary gland, saliva, salivary amylase. The mouth receives food, tongue help in swallowing, manipulating the food for chewing and in perceiving taste. The teeth subdivide the food for easier digestion. Three salivary glands, parotid, submaxillary, sublingual, secrete saliva. Saliva helps in swallowing the food by lubricating the food, and neutral pH of saliva prevents the decalcification of teeth and keep the mouth clean. In mouth, enzyme named salivary amylase or ptyalin is present. It starts the digestion of starch by breaking into dextrins. Dextrins are the polysaccharides. The secretion is controlled by nervous reflexes. Digestion in stomach. Stomach has gastric juice, HCl, pepsin, lipase, renin. Stomach is J-shaped organ and secrete gastric juice. Gastric juice has HCl, an enzyme pepsin. Pepsin gets secreted in an active form called pepsinogen. Lipase also gets secreted in inactive form. Cause of acidic nature of gastric juice. Renin is only present in infants and it digests milk protein casein. HCl having pH of 1.2 to 1.5 activate the inactive pepsinogen to active form pepsin, and HCl denatures the food protein for easily digestion. Germicidal effect of HCl prevents the microorganisms, and protein digestion starts in stomach by pepsin, and pepsin converts protein into proteoses and peptones. Pepsin is endopeptidase as it hydrolyzes peptide bond within the main polypeptide structure. Pepsin act on peptide bond formed by amino acid with aromatic ring or decarboxylic amino acid with 2 COOH group. Pepsinogen with the help of HCl converts into pepsin and peptides. Pepsinogen again converted into peptides and pepsin, and food proteins get converted into proteoses and peptones. Who controls the secretion here? Gastric secretion are initiated by nervous plexus. Continued secretion is controlled by gastrin hormone. Histamine, the decarboxylated amino acid, and histidine is also to stimulate gastric juice production. Role of pancreas in digestion. Pancreas secrete pancreatic juice. Pancreas secretes 600 to 800 ml of fluid. It is alkaline in nature. pH is equal to 8.0. It contains organic and inorganic substances. The enzymes are trypsin, chymotrypsins, elastase, carboxypeptidase. All are secreted in inactive form called zymogens. The other enzymes are amylase, lipase, cholesterol ester hydrolase, phospholipase, ribonuclease, deoxyribonuclease, and phosphodiesterase. Sodium chloride and bicarbonate are major inorganic constituents. Zymogens are enzyme molecules synthesized but inactive form. When food enters the stomach or intestine, the inactive form gets converted into active form by HCl. Pepsinogen gets converted into pepsin. Trypsin is secreted in an inactive form, trypsinogen, and enzyme enterokinase convert it into active form, trypsin. It acts on protein, protease, peptones and convert it into polypeptide, peptide. It attacks the peptide linkage containing arginine and lysine residues. Trypsinogen gets converted into trypsin and hexapeptide. Proteoses plus peptones gets converted into polypeptide and peptides. Chymotrypsin. Chymotrypsin is secreted in inactive form, chymotrypsinogen, which is activated by trypsin. It is specific for peptide bond containing amino acid residues. Elastase in active form, proelastase, is activated by trypsin to its active form elastase. Elastase attack peptide bond next to small amino acid like glycine, alanine, serine. All three enzymes like trypsin, chymotrypsin, elastase are endopeptidase. What is endopeptidase? These are the peptide hydrolases. These are the peptide hydrolases that hydrolyze centrally situated peptide bonds. Carboxypeptidase. In active form, procarboxypeptidase gets activated by trypsin. Carboxypeptidase that attacks carboxy terminal peptide bond liberating single amino acid. So here it is to note that carboxypeptidase hydrolyze peptide bond from the end of the peptide bond. Therefore, it is called exopeptidase. This enzyme acts on peptide bond of those peptide chain that contains free COOH group. It is also called carboxypeptidase. Trypsinogen gets converted into trypsin and hexapeptide. Then trypsin further converts chymotrypsinogen to chymotrypsin plus peptide, and trypsin again proelastase to elastase, procarboxypeptidase to carboxypeptidase. So all inactive form gets converted into active form. So first trypsinogen itself converted into trypsin, and then this trypsin converts every active form into active form. Amylase. Pancreatic amylase break down starch and glycogen. The hydrolysis of starch and glycogen produce maltose, maltotriose, mixture of branched oligosaccharides, non-branched oligosaccharides, and subglucose. Lipase. Pancreatic lipase hydrolyzes the primary ester linkage. The presence of bile salt help in emulsification of fat, increasing fat digestion. 2-monoglyceride and fatty acids are the end products of fat digestion. 1/4 of triacyl glycerol is completely broken down to fatty acid and glycerol. Cholesterol ester hydrolase act on cholesterol ester to liberate free cholesterol and fatty acids. Phospholipase break down to phospholipids. Ribonuclease, deoxyribonuclease, phosphodiesterase. The above enzymes are responsible for degradation of dietary nucleic acid, ribonucleases, and deoxyribonucleases secreted by pancreatic juice, hydrolyze RNA and DNA to oligonucleotide. Oligosaccharides again break down into 3,5-mononucleotide by phosphodiesterase. Nucleotidases remove the phosphate group and release nucleoside. These are absorbed by mucosal cell and further degraded into free bases before the uptake. What controls the secretion? Secretin, hormone produced in the duodenum in response to chyme, the complex content of stomach. Secretin causes the secretion of protein-free electrolyte solution that has high concentration of bicarbonate. Pancreozymin, cholecystokinin. Pancreozymin is hormone released from the mucosa of duodenum that causes the release of rich juice from the pancreas. Role of bile in the digestion. Bile is produced by liver and stored in gallbladder. During digestion, liver contracts and supplies the bile through the duodenum. Bile produced in the liver is less concentrated than bile stored in gallbladder, and it is high in bile salts, cholesterol, and pigments. Bile acids are cholic acids and deoxycholic acids, and salt of these acids with glycine and taurine are called bile salts. They have lower surface tension and prevent coming together of small fat droplets in the intestine and allow rapid digestion of fats. This is called emulsification of bile. Hydrotrophic action of bile. Bile acid combined with fatty acids and help them more easily absorbed. The presence of bile in the intestine is to accomplish digestion and absorption of fat and also fat soluble vitamin. Digestion in the intestine. Intestine secretes 2 to 3 liters of alkaline fluid. They have both organic component and inorganic component. Sodium bicarbonate and sodium chloride are inorganic component, and organic includes enzymes such as aminopeptidase, dipeptidase, disaccharides, phospholipase. Enzyme of intestinal juice. Aminopeptidase is an exopeptidase and it cleaves the peptide bond next to N-terminal amino acid of polypeptide and oligopeptide. Dipeptidase completes the digestion of dipeptidase to free amino acid. Control of intestinal secretion. The specific disaccharides and oligosaccharides like maltase that removes single glucose residues from alpha 1,4 linked oligosaccharides starting from non-reducing end. Isomaltose, hydrolase, 1,6 bonds in limit dextrose, beta-galactosidase remove galactose from lactose and sucrose hydrolyze sucrose to glucose and the intestinal juice contain phospholipase that attacks phospholipids to produce glycerol and fatty acid, phosphoric acid and bases such as choline. The secretion is stimulated by enterokinase produced by jejunum, which in turn released by entry of acid chyme from stomach to duodenum. Thank you so much and study hard and wait for the next section. I hope you all are safe and stay protected and stay safe. Thank you so much. Bye-bye. Take care. I hope you do well in exam. Bye-bye.